anche se in realtà non è che...
Se sarebbe quella casa se non...
È arrivata.
Perché possiamo studiare molto anche
Come no?
Il motorino evidentemente è veramente lenta, cioè.
C'era pure traffico. Perché c'è la diretta streaming, quindi devi per forza usarlo. Cioè, fosse stato solo internos. No, no, dai. We are ready if you want.
Okay, so good morning. This is the, I would say, last day of full lecture of this training school on microbial technique applied to hydrogen research. We will have a series of talk which will go more, I would say, directly to the application and to the methodology part after the previous day where we have been settled with the principle and the base and foundation of the microbial science. And today we will have a first introduction by Annarita Ricciardelli. Then the second talk will follow, which will be 1 hour. There will be a second talk later at 11 till 12. And usually we have half an hour of discussion, conversation, interaction to that. Okay, so I'll leave the word now to Annarita, and which I thanks for the presentation. Thank you very much.
Thank you for inviting me here today. It's a pleasure for me. And I'm a postdoctoral student, let's say postdoctoral researcher at University of Naples Federico II in the Department of Biology. And today I will introduce you in the microbial world by talking about microbial techniques, focusing on those that are culture-based approach for the study of microbial communities in hydrogen-related research. So this is just an overview of the microbes can be defined as hydrogen producers that are basically widespread in all the domains of life, so both in bacterial, archaeal, and eukaryotic domains. And they can produce hydrogen and release it in the environment through different metabolic pathways. But then we can also have hydrogen consumers that I think you understood well in the previous lectures. that use hydrogen to, let's say, reduce carbon dioxide or sulfate in their metabolic pathways, so methanogens, acetogens, and sulfate-reducing bacteria. But where we can find these microbes in the environment? So basically, environments where we can have anoxic conditions, but especially those in the geothermal environments like hot springs, hydrothermal vents, both shallow water or deep sea, in the anoxic part of the marine sediments, but also in the subsurface area. First, before starting to sample and then to characterize the microbial communities in these environments, we generally start asking some general questions like who's there, what are they doing, who is doing what, and how they are doing this, and which which extent, let's say. And these questions help us to understand the environmental conditions where these microbes live, and also the type, let's say, of the lifestyle of microbes that lives in this environment. So these questions help us to define the study, and then we can move to the sampling part, so sample collection, or what is coming after the sampling collection, so how we want to preserve both on fields and when we move back to our laboratories, basically. How we want to process, so analyze these samples, and how we want to, let's say, interpret this data, and the subsequent results. First, let's say that sampling for microbial biodiversity can be very different depending on the environment where we are going to sample. You can stay, for example, in the Puna plateau, sampling hot spring, terrestrial hot springs at 3.5 kilometers of altitude, or you can go sampling, for example, hot springs in Iceland, where the weather is always cold, rainy, and windy, or maybe you have to dive or snorkeling to sample from shallow water hydrothermal vents. Or you can also stay like for weeks or months in a drilling site, waiting for the cores coming from the subsurface to come out. So all the environments requires also our skills that can be different from each site. The first thing that we generally do when we reach the sampling sites is collecting environmental data. So first, the, let's say, geographical parameters like latitude or longitude to exactly know which is the location of the site that we are sampling, also for a future reproducibility of the sampling. But then we are collecting these parameters that are like fundamental for us to understand like in which environment these microbes that we are going to characterize, they live. So temperature, salinity, using a specific, let's say, instrument, but also sometimes we use this multi-probe that basically putting the probe of this instrument into the fluids, for example, coming from a geothermal SIP. We can collect also like pH parameters, dissolved oxygen concentration, if there's a carbon dioxide release, all, let's say, all parameters that shape the environment where microbes live. Now, for the practical sampling, basically, we usually collect sediment samples and fluid samples of, in this case, this is a terrestrial hot spring. that we then both characterize in terms of microbiological, let's say, point of view, but also geochemical point of view. And we collect also a background sample just to be sure that all the microbes that we see in our samples are not coming from contamination from like human or environment contaminations. So for sampling fluids, we can like use different kind of, let's say, techniques, but also instruments. The easiest way is to collect fluids seeping out, for example, from an hot spring using sterile containers. So basically containers that we sterilized, so they basically will contain only the microbes of our interest. But we can also use a pumping system, let's say, because first we can collect a huge amount of fluids, but also like specific fluids, for example, that are seeping out from a geothermal spot. So using those kind of pumps. Si vede? Bene. You can directly put the tube of the pump into the spot from where hydrothermal fluids, for example, are seeping out. And you, in this case, you can be sure that you're just collecting geothermal fluids and not, for example, seawater that can contaminate your geothermal sample. But we can also collect fluids in other ways, depending on the kind of sample that we are collecting. For example, if we are in like in a mud pool, in this case, it's difficult to collect only fluids separated from the sediments. So we can collect all the mixture, let's say, and then we can separate the fluids from the sediments, for example, through centrifugation, which is basically an instrument that with this high-speed rotating rotor separate the sediments that are basically going down into a container and then leaving your fluids up. If we are interested in sampling the biomass that is contained then in these fluids, generally we use filtering systems. These filters are based on a membrane that have specific cutoff of their pore and can basically let through the application of a pressure, for example, a syringe or a pump directly, you can basically let the fluids pass through the filters and the biomass will be stuck onto the surface of the membrane. Of course, depending on which microbes you want to sample, if you're interested in viruses, bacteria, or bigger eukaryotic cells, you can choose a different cutoff of these filtering membranes to selectively sample your microbial communities. We can also sample microbial biomass that is basically aggregated in this community that are called biofilms, that are basically sticky or fluffy sometimes. Let's say carbohydrate-based matrix that you can directly sample, for example, like swabbing, so using these swabs like cotton stick directly from the site, or you can take all the biomass with the syringe or with some instruments that are called push corer, that are basically sampling both the mat, let's say, microbial mat that is formed on the seabed surface in this case, and also with the use of scrapers, so scrubbing the surface. Sampling sediments instead generally requires the use of these coaters. So basically, these cylindric containers that you can directly push or grab into the sediments and catch all the layers depending on which depth you want to collect your sediments. The balers are very simple, are generally plastic based for shallower, let's say, sediment layers, but you can also use like more complex systems like the downhole pressure vessels that are generally preferred in deeper subsurface environments where the pressure is higher. Then we also have drilling systems like for the continental part, let's say, that is basically are designed to penetrate on the Earth's surface and just reach the material that you want to extract. And it can be a very, very easy and simple, like an end auger for generally used for soils or for soft sediments like sands. Or can be very like a more complex system, like a rotary drilling rig or a diamond core drill that are basically going more deep into the crust, let's say Earth crust. Also up to several kilometers. But we can also sample like these sediments, also very, very deep sediments at deep, let's say, ultra deep water sediments using this drill ship that are basically endowed with this drilling system that works offshore. The most important thing that we have to take in mind, so we have to focus on, is that coring and drilling allow us to collect these huge, also in some cases core sediments that basically let both the sediment stratification, but also the microbial distribution into the sediment stratification intact. So this is the best part of the drilling. Then when we have our sample, we can store them generally by refrigeration or freezing, depending then on which kind of analysis we want to carry out downstream. So generally refrigeration is for culturing or for isolation of the microbes. Whereas if we're interested only in analysis on the DNA or on the chemicals that we can find in our samples, we can also freeze our sample at -20 or -80. For the analysis of our samples, we have a lot of different techniques that can be sort of classified as culture dependent or culture independent based on the use of culturing techniques. But we can also have different techniques that basically change on the type of information that give us. So can be qualitative, quantitative, or semi-quantitative. But we can also refer to the analytical principle that these, let's say, on which these techniques are based. And we can have like chemical, biochemical, molecular analysis. Geochemical analysis for us are very important. I'm pretty sure that you know better than me what it's a geochemical analysis, but in general, on our sample, we are used to perform ion chromatography to have information regarding the major ions that we can have in our fluid or sediments, let's say, and ICP-MS to have information regarding the metals that we can find. Let's focus on the ion chromatography. Basically, I just reported the plot that maybe you already know. This is a Piper plot that basically tells us let's say, the different based on their interaction with the rocks. So, for example, when you see calcium sulfate waters, you can you can say that are typical, for example, of gypsum groundwaters or mine drainage, or waters that have interacted with volcanic gases or that are rich in sulfur. What we see is a sample that can contain sulfate reducers or sulfur oxidizer bacteria. Same, for example, in sodium carbonate waters, you can say that are typical for deeper groundwater. I can say that it's a kind of environment where we can find methanogens, so carbon dioxide rich fluids. Same, of course, in case of cations or in case of anions. Regarding instead the ICP-MS data that we have, so basically the information regarding the metals, what we, let's say, understand from this data is the possibility to have some metabolic pathways, because transition metals or trace metals are fundamental components of the enzymes that most of the microbes use for their metabolic pathways. So finding I don't know, let's say molybdenum and nitrogen ions, for example, in our samples, can let us predict that it's likely for us to find nitrogen oxidizer or nitrogen reducing bacteria. Now let's move to microbiological analysis, and for microbiology microbiology characterization, we can use culture-independent and culture-dependent approach. They are both valid, but they say us different things. For example, culture-independent approach allow us to understand the microbial diversity, avoiding the culturing that in some cases can be a bottleneck, because not all the microbes that we can find in the environment are culturable. Whereas, of course, culture-dependent approach can give us more information regarding all the microbes that we are able to cultivate. But let's say that a successful approach is generally a combination of the two techniques. As regarding the culture-dependent approaches, first let me just give you some basic informations, like what is microbial culturing. So basically it's a process of growing microbes in specific and controlled environments. So basically we prepare what is called the culture medium, that it's basically a broth where all the nutrient substances trace elements are dissolved, and it can be liquid but also solid, and basically all the nutrients are required for the growth of the microbes that we collect from our environmental samples. The concept of sterile, of course, is fundamental, because in this case we can only let the microbes that we are interested in to grow, and not also the possible contaminants coming from the environment. We can have the solid medium poured into a petri dish. It's called petri dish, and in this petri dish we can have a streak, let's say, of the microbial cultures. The process of putting, practically putting the microbes into the culture medium is called inoculation. And then after inoculate the microbes, we let them grow under these under like their preferred conditions, and this it is called incubation. At the end, after the incubation, on a petri dish, we can find single colonies of the microbes that are basically a group of microbes coming from a single cell that have has multiplied and become visible as a spot. So, culturing can be performed by different techniques. So one of these is the enrichment culture techniques that basically is based on the use of selective conditions. So a culture medium where all the nutrients are selective, for example, for a niche. So for a particular ecological niche of microbes, let's say that we want to have or maybe like isolate only hydrogen consumers from our environmental samples, we can set up a precise medium in which only hydrogen consumers can grow. Or we can perform microcosm experiment, but I'm not going so deep in this because my colleague can better show you this kind of experiments. So basically it's just a sort of reproducing a smaller scale of the environment of the microbes where you isolated them. And then there's the pure culture technique. In this way, we can isolate just one species from all the microbes that were present in the community. So basically, the sample is spread on a petri dish, and from all the different colonies, you can pick just one. And streak it with a specific dilution pattern on the petri dish, and then after the incubation you can find the single colonies on the petri dish, on the solid medium. We have also different let's say, way to measure the microbial growth, depending on also which kind of culturing techniques we are working with. The most used is the evaluation of the optical density or the turbidity measurements. So basically, when you inoculate your microbes in a liquid medium, the growth of these microbes increase the turbidity of the liquid medium. And this turbidity can be measured through the use of a spectrophotometer, because the light interacts with the matter, basically, so with the biomass that is present in your liquid medium. And based on the amount of the light that is absorbed or scattered, let's say, we can like measure what is called OD value, so optical density values, that is proportional to the microbial growth. Then we can also count the let's say the colony, so measuring what is called the CFU, so colony forming units, because starting from a single cell, then on the petri dish, you can have like as much colonies, as much single cells were present in your sample. And we can also have, of course, the first two are, let's say, quantitative, but we can also have semi-quantitative, like, for example, the dry weight measurements, because the increasing biomass, of course, can be related to an increases weight of the pellet that is, of course, proportional, but doesn't say you, of course, how many cells. It's just like an estimation. Another, let's say, semi-quantitative can be the microscopic count that can give you information regarding, for example, 10 microliters of your sample that you put on a glass slide that you put under the microscope, and you can estimate the total amount of the cells, counting the cells that are present in those 10 microliters. Now, we have always the problems of the unculturated bacteria because, of course, especially when we are, let's say, sampling in deep sea hydrothermal vents where the temperature is higher than 100 degrees and the pressure is different from those at sea level, it's like a little bit difficult for us to reproduce these conditions to grow these kind of microbes in a laboratory scale. So in some cases, where it's like the culturability of the microbes, it's a gap that in case of like studying unculturable microbes doesn't give you information. And also pure culturing, so the isolation of the pure culture is unfortunately the only official approved approach that can lead us to the description of a new microbial species. All the enrichment or metagenomic that you will see soon are not approved from what is called the International Committee of Systematic Prokaryotes, that basically publish all the approved new species name on this International Journal of Systematic and Evolutionary Microbiology. Of course, being uncultured, let's say it's an operational definition, and it's not really a feature of the organism because it's like it's because of our possibility are restricted, so that's why they are unculturable. And the way that they use to classify, it's not really, let's say, polite for us. It's defined as microbial dark matter. So I think that maybe this needs to be changed. Let's move to the culture-independent approach. I will just give you some example of microscopic techniques. For example, we can use fluorescent microscopy to visualize the cells under the fluorescence microscope using different fluorescence let's say, or probes. For example, if we want to count every single cell that is present in our sample, that maybe it's like I complex, let's say, we can use a specific stain that is like going to stain all the cells without discrimination between all the function, lifestyles, and morphology. Whereas if we want to discriminate in a sample, if we have like viable or dead cells, we can use another kind of stain that it's like discriminating between the living and dead cells. Or this is the most, let's say, interesting part. If we are interested in some metabolic pathways, let's say, we can probe specific genes involved in these metabolic pathways, choosing different stain. And in this case, we can have images like this. Also of complex community without literally isolating cells from a biofilm, let's say. Also, in this case, confocal laser scanning microscopy can give us information about the, let's say, stratification of microbial community in biofilms or mats. And it's basically collecting two-dimensional images, and with this option that is called Z-Stack, it's basically reconstructing the three-dimensional image. Also, flow cytometry, it's generally used to, let's say, switch or selectively count the cells on the basis of their shapes. But it can also be used using the FACS, so it's a fluorescence activated cell sorting with the use of specific probes. So we can also discriminate between cells depending on which probe we use. So which, let's say, subpopulation we want to highlight. Also, we can measure the growth or the activity of microbial community by measurements, by measuring the specific metabolism. In this case, is represented the sulfate reduction measurement that it's based on a colorimetric spectrophotometric assay. And basically, you can derive from this spectrophotometric assay the concentration of the sulfate that is basically reducing in time and with the formation of hydrogen sulfide, but also the consumption of the lactate that these kind of microbes use as electron donors. For the moment, I'm done, but I'm pretty sure that you know that is something that is missing. And for this, I will give the microphone at my colleague, but maybe after a break, if you want. Questions? Tutti? Green? Cells. Sì. Bacterial cells. Under the microscope, you cannot really see, depending on the magnification of the lens that you use, of course, you can see the cells as very, very small dots or very huge or going in deep into the cellular structure, depending on the microscopic technique that you use. In this case, the DAPI staining, it's basically a stain that is penetrating into the membranes of the livings of all the cells, let's say. And so basically you can have a vision of all the cells that you can have in a sample. In the other case, where you see red and green spots, the red spots are the living cells because it's cyto9 in this case that is penetrating into the membranes of living cells, whereas the PI, that is Propidium iodide, it's another kind of strain that it's basically its structure is not penetrating into the membrane of living cells. So basically just attaching to the DNA of the dead cells. So in this case, we can like discriminate between them. Questions? Regarding the microbiological point of view, not really. And actually, sometimes it can also happen that you, especially when you're sampling, I don't know, seawater has a concentration of cells that it's not huge, let's say. So you have to maybe pass through the filter like also 10 liters of seawater, but also it's very common also for geothermal fluids. So in this case, it can happen that when we go back to our lab and we start to like analyze our microbiological sample, it can happen that the amount of the biomass that we collected is not really sufficient for us to have good quality information. So it can happen, yes. Sì. Yes, it can be, you're right. And that's why we collect always like different kind of samples, let's say. If we want to isolate living microbes, we try to of course, store these samples at +4 degrees. In this case, generally cells are like sort of quiescent. So it's not really changing the abundance of the relative abundance of the species into the sample. But then it's also true that if we sample, for example, in Argentina, and we store our sample with these icy cubes, maybe the temperature is not really 4 degrees. So it can be, even if when you sample, let's say, at 70 degrees, 4 or 10 is not really changing. If you store them at 4 or 10 degrees, it's not really changing, let's say. But then to have a precise information of what's exactly the relative abundance of that species exactly in the moment when we sampled, we freeze also the same sample. So from that, we can then apply culture independent techniques that you will see very, very soon. Whereas for culturing, or maybe you just want to isolate, I don't know, one species or two species from there, and then even if it's just changed, the relative abundance is not really that one species can die and the other one can dominate. Like the timing, it's always like compared to the first day of the expedition to the day that we go back in our lab, it can be 20 days, generally. But it can also be in the same day. For example, when we come here to sample in shallow water in Pozzuoli, basically during the morning you collect the sample, and the afternoon you're already manipulating your sample and analyzing them. You're welcome. I think so. I didn't check, to be honest. But yes, generally it's the best way to visualize, so microscopically visualize the different microbes with different lifestyles in the same community. And you can probe each specific metabolism with a specific stain. So you can see methanogens in blue, methanotrophs in purple, and sulfate reducers in green, let's say. Stratifications, yes. Can be very nice, yes. Other questions? Okay. Thank you. Do you want a break or just continue? Fine. tempo per le domande.
Okay, are you guys ready? Okay, so hi, I'm Deborah Bastoni. I'm a PhD student at the University of Naples Federico II, and we will continue on the topic of investigating microbial diversity, but focusing on culture-independent approaches, okay? I don't know how much you guys know about microbiology, so if you don't understand something, if you have questions, stop me, we can like go through it better, okay? So, as you saw first, we have many ways to investigate microbial diversity. When you go and sample a site you're interested in, you can choose many ways to look at microbial diversity, right? But we have a big problem when looking at microbes, which is that most of them, a big part of them, are not easily cultivatable in lab. So it's like you want to study botany, but you only have the plants that grow in your backyard garden, okay? It's a very limited amount, so you cannot really understand a lot about it if you don't also go through all those ones that cannot grow in the lab. So we had to develop a number of ways to also study all those microbes that don't like to grow in our labs, right? And this is where culture-independent approaches come in hand. So thinking about the way Anarita explained our sampling approach, you choose one site you're interested in, you go there, and you see your microbial community. You can actually, as you see in this photo, sometimes you really see it. And you can like ask yourself two main questions, right? Who is there and what they're doing, okay? And to answer to these main questions, we have two different approaches, two techniques. And to answer to who is there, what's the community like, what's the structure of the community, we have taxonomy. Taxonomy helps us to identify the microbes that are present in your environment. So if we go back to the analogy of your garden, with taxonomy, you understand that you have one lemon tree, two apple trees, and one tomato plant, okay? With taxonomy. So you can get the list of all the types of organisms you have in your garden, and also the amount of organisms. So you have two apple trees, three lemon trees, and whatever, okay? This is what taxonomy does for your sampling information. And then we have functions, which is what those microbes are doing. And information about the function of microbes can tell us a lot also about the way they interact with the environment, because functions means that they are actually doing some metabolic processes. So they're performing biochemical reactions that interact with the environment. They use something, substrates from the environment, but they also produce molecules, for example, that are going to change the geochemical environment they are in, okay? So we need to combine these two informations to get a very good and deep understanding of the community in your site. So let's start with taxonomy. So as we said, taxonomy tells us who lives in our sample, right? So as you see here, we have a site, and you can get all of this information about the phylum of your microbes, up to much more detailed information about their phylogeny, okay? To understand the taxonomy of your microorganism, we use marker genes, okay? Marker genes are very useful in taxonomy because they are genes that have been chosen because they're very recognizable, so easy to find in a DNA, and they usually are ubiquitous, so you'll find them everywhere, okay? So you find one gene which is available in all microorganisms, and it's very easily recognizable. The most used marker gene for taxonomy microbes is the 16S. This gene is very well conserved. Conserved means that it has some parts that are always the same. So if you go to a DNA, you can easily find the 16S, and you're sure that's the 16S, okay? And then it has some parts that vary a lot. So you have, imagine one piece of DNA, you have some parts that are always the same, which means they are conserved across all microbes. And then you have some parts that change, and they change at different rates, okay? Looking at how different these variable parts are, you can understand how close or how different microbes are. And you can like give a name to them, okay? So let's say, doing an easy example, you have a wolf, a dog, and a cow. Looking at those variable parts, for sure, a cow will have a much more different part than dog and wolf, right? Because they change much more. And so using 16S, we can do a lot of things. We can build trees of life, so we can understand how different and how close or related microbes are in our samples, or we can even identify which microbes are present in our samples, okay? So let's make an example on your case of study. So you're interested in hydrogen, like hydrogen-related microbes, right? For example, you look for a hydrogen-rich site, okay? And you wanna identify stuff like hydrogenotrophic methanogens, right? So you know their name, you do the 16S on your site. So you go on your site, you collect the sample, then you bring it to the lab, you freeze it. So nothing changes in the site, okay, in the sample. The community stays how it was when you sampled it. You bring it to the lab, you extract the DNA, and you only sequence, so you only get the sequence of the 16S. And you're ready to do your taxonomy. You can do something like this, for example. So on the bottom, you see all those, these ones. These are all the sites, so you can compare different sites. And in different colors, you have all the specific groups we identified with the 16S, okay? So you can see, like, the bigger the colored piece, the more abundant is the group. And this is very useful because you can even identify those specific groups you're interested in, okay? You can look for the name of the microbe you're looking for, and you can compare how abundant it is between one site and another, for example, okay? You can even do something like this with 16S. So you can choose also a specific part, a specific group of microbes you're interested in, and you can see like the dimension of the bubbles, it's related to how abundant it is, okay? Across different sites, how abundant is that specific group. And then with 16S, you can do a lot of other very interesting things. For example, this is alpha diversity. Alpha diversity, you guys know what diversity means? Yes, okay. So, for example, you can like see how diverse is the community between one site and another. And this is very useful when you're also considering environmental parameters, like, for example, temperature and pH. If you think of a site with very high temperature and one which has mid-temperature, which one do you think has the higher diversity? Yes, because like less organisms are like adapted to live in that high temperature site, right? And you can even do stuff like that. Have you ever seen one plot like this one? Okay, so we said that with 16S you get the abundance of each specific group in your site, okay? So when you have multiple sites, you can calculate like the distance, like a number, which represent how different is your site between another, okay? And you can then plot in a bidimensional space. And as you can see here, you have like different sites, and the closer they are, they are more similar in terms of microbial community. So if we go back to that garden example, if I have, I don't know, 3 apple trees and 3 lemon trees, and my neighbor has completely different garden, probably here they will be very far apart in this plot. But if my neighbor has the same plants or very similar, they will be closer, right? And if we color this plot with environmental variables, for example, here you see with temperature, we can see very cool trends if that variable is influencing the diversity. As you can see here, we have on the right of the plot, yes, the red part, we see all those high temperature sites, which probably are very similar in terms of microbial community, right? Because we have the same groups that are adapted to high temperature, so they're very similar. And then if you move to the left, we shift in temperature and go to lower temperature sites, okay? So with 16S, we still, we can get a very good understanding of how the community interacts with the environment, okay? So it's not telling us, it's just telling us who is there, okay? We can still imagine what is happening based on the knowledge we have on some specific groups, right? So you see that one microbial group is very abundant, you can still think, okay, I know this group does this, probably this is happening, okay? But you don't know for sure. While going into function will tell us specifically what are the processes, the biochemical reaction happening and the metabolism that are active in that place. So yes, taxonomy has major advantages because you can use very easily recognizable marker genes. They're spread across microbial groups, but we cannot get information about what is happening. So we have to switch to functions. So the question here is what they are doing. And to do functional analysis, we can move through different steps, and we have different layers of information. Based on the layer of information we use, we can use genes, so DNA, we can use transcripts, or we can even use proteins, okay? So depending on the approach, here is a trade-off of information. You can get a lot of information with this kind of analysis, but lower preciseness, or you can get very deep into the information, but having less information, losing information across, along the way. So it's always a trade-off of which technique you use here, okay? But let's understand better these layers. So we said we have DNA, RNA, and proteins, okay? So if you think an IKEA shelf, okay, you need to build an IKEA shelf, you go to IKEA, you choose your shelf, and you go back home, you open the box, and the first thing you see is the manual of instructions, right? You go through the manual, there are many things written, you don't necessarily need them all to do your shelf. So that's the DNA. That's the whole manual of information. Whatever you need to build, whatever you want, it's in there, the DNA. Sometimes even more stuff that you don't really need, okay? That's your IKEA manual of information. So you choose which part you want to build, and you start reading it. You start checking whatever you need to do. That's the transcription, that's the RNA, just the part that you want to build, okay? That's the RNA. And then you actually build that shelf. That's the protein. So you have the information written in the manual with among all other stuff you don't need. That's DNA. Then you go on, you see the transcript. So what you actually read, what you actually need to build your shelf, and your shelf, which is the protein, okay? So let's start with the most I would say superficial. It's not superficial, but the first layer, which are genes, so DNA, the manual of instructions. Here you'll find all the genes available in your sample or in your organism, okay? Depending on what you're looking for. The fact that you have all those information doesn't necessarily mean, as in the IKEA methods, you're not building it, right? You have the information, but it's not necessarily happening in that moment in that community. So when you use DNA, you have to remember that that's only the potential of the community. It doesn't necessarily mean that those processes are happening in that specific moment, okay? So looking at the genes and DNA, you have two different approaches you can use. One is very specific and targeted to your hypothesis, let's say, and one is more general and gives you an idea of the community and like the whole like potential of the community, okay? One thing you can do is look at target genes. So let's say you're interested in hydrogen metabolism, you look for hydrogenases. So you just check for the sequences of your specific genes that you're interested in. You look across all the DNA of your sample, you just record and select the hydrogenases, and you build a table that tells you how many hydrogenases are available in that sample, okay? And you can get information like this one. So on the bottom, you have many different sites, you see here. And then on the left, those are the specific genes selected. And you can see how their abundance change across different sites. Okay, so you are just selecting those genes of interest for you. And you don't know the species here. You just know that in that sample, you can still do it on a specific organism here. So if we go back to what Anarita said, you can select one specific organism using culture, and then you can do this. And you know that in that species, you have some specific genes abundant or not, okay? But so you're just looking at the abundance of the genes. You don't know who owns them, okay? Otherwise, you can check the whole DNA. And this is what we call metagenome. So you check the samples, you extract the DNA, and you select the whole amount of DNA of the whole community. You don't even you don't look for specific genes. You have all the genes of that community. And you can do very interesting things like this one. So you can select and classify specific genes for their metabolism. So at the end, enzymes are involved in sulfur cycle, nitrogen cycle, carbon fixations. You can get a very broad information about the potential metabolism happening in that community, okay? And still, this has to be always referenced to the environment you're looking at, okay? So checking the temperature, the pH, the geochemical information about that site with all these amount of data about the metabolic potential, it can give you a lot of information about your microbial community, right? But this is still a lot of information to process, a lot of data. So that's where the trade-off comes in. Here you have a huge amount of data to process and to understand, while in the other case, you have a very specific selected hypothesis. You can look for the specific answers to your question, right? And usually using both techniques is very useful, okay? So we looked at genes, and as I said many times, that's just what the community could do. It doesn't mean they are doing it, right? And we go into transcripts. So you have your manual, you choose what you want to build, and you start reading it. That's the RNA. So the genes that are actually expressed, okay? So they are being written, read, and your body starts to want to build them, okay? You're not building it yet, you're just reading it. So in other words, the genes represent the potential. Transcript shows what microbes are actually doing in response to an environment. And you can get information like these ones. So you can analyze the transcript of the specific genes you're interested in, for example, for two specific sites. And you can probably, if you're interested in hydrogen metabolism, you can, for example, check the amount of hydrogen in one site and in one other, or other environmental information, and see how much of those genes is actually expressed in one sample compared to the other one, okay? So here you have a more specific information, but you have to remember when we go from one layer to the other, it gets more complicated. So one thing is extracting DNA, one other thing is extracting RNA, which is much more difficult, and so you have much more precise information, but you can lose information along the way here. Okay, so you have to remember these things. And then we can go into proteins, so the actual shelf that you have already built. Okay, so the fact that you have RNA and you have the transcript so that you read the instruction doesn't necessarily mean that you were able to build the shelf for many reasons, environmental conditions, whatever. So if you have the proteins, you're sure that something is really happening. If you don't have the protein, but you only have the transcript, it's likely to happen, but it doesn't necessarily mean it's happening, okay? So the proteins are the end products of gene expression, and they perform all those biochemical reactions we're interested in that are influenced and influence the environment, okay? So you extract, you have your sample, you process it, you get the proteins, and then you can do proteomics. So you can do the analysis of all the proteins in your samples. Still here, you can choose to look at specific proteins that are involved into your question and hypothesis, or you can look at the whole diversity, okay? And proteomics give us direct information of the functional activity in your sample or in your specific community, right? So let's say you have hydrogenases transcript, so you have the RNA. It doesn't always translate to active proteins. While if you have hydrogenases proteins, you know that those microbes are actively doing, like, metabolizing hydrogen, okay? But still, like, you go up to proteins, this is much more difficult. You lose other informations, you go deeper and deeper into this hole of, like, hardness and okay, so doing a small recap, you have genes that tells you the whole potential. Then you go into transcript, which is what your genes want to express. Then you go into proteins, which is actually what they're doing, okay? So you have your final shelf done, but there is one even, like, deeper layer, which is going to look at how your shelf is built, how is it working, is it sturdy, working well? And that's looking at real-time activities of your community, so metabolic rates. So measuring reaction rates allow us to understand if those proteins are working well, how fast they are working, what they're doing, okay? Okay, and for example, two techniques we can use here. We can, for example, probe a substrate, in this case, hydrogen, and we can see how fast it is incorporated into, like, by those microorganisms, and this tells us how quickly they're working. Otherwise, you can do also respiration assays, so monitor the consumption of substrates and how fast they are producing end products of their metabolism, for example, methane or CO2, okay? And this tells us how fast these microbes are working. So let's say you have two different experiments of two different samples. One has completely different substrates to another one. You can see what are the end products of the reaction of these microbes, how fast they are working, if different temperature can influence the same reactions, right? And for example, here you see the hydrogen concentration against the rate of hydrogen consumption. So this tells us a little bit about what the community is, how fast the community is working, okay? So, like, in conclusion, you have taxonomy that give us information about the community, who lives in our site. And then you can go in asking, what are they doing? You can use genes, which are very like give us a broad information about the potential of the community. You can look at transcript, which is a snapshot of what they are expressing. Then proteins, which are like really what the key reaction that have been like that are happening. And then you go into reaction rates, which is a direct measurements of their activity in that moment. Okay. And that's it. It was very quick. If you have questions, guys. This one. Okay. Well, this is like just an example. It could be whatever. Like you can measure the production, let's say, of methane. You can measure methane across time. You have your community. You measured methane production at day 1, day 20, day 60, and you see how the curve is growing based on your on timing, on temperature. You can set up an experiment where you have one community, another one. You keep one at 10 degrees, one at 60, and you measure how fast they're producing methane, for example. Okay, for example. Otherwise, you can measure the amount of substrates you have available, okay? We did an experiment looking at the amount of sulfate available in that site, and we saw sulfate growing, so someone was producing sulfate in some. Okay, this is just an example to show you that you can measure not only the community, but also what they're producing and using. Okay, sorry. Well, this depends if you're measuring in an experiment or if you're measuring in an environment. So in environment, you have a complete community. And what happens usually in a stable community, that you have someone who produces something and someone who uses. So you have a community that always producing and making available that substrate, and someone who is using it. So it's always available and someone is using it again. While in experiment, you have that defined amount of something, and once the community has used it all, it is done. So you probably will reach a steady state, and then your community will start decreasing because the food is ended. Okay. Thank you. Yes. This one. These are 3 different campaigns. So they're comparing different sites from different campaigns. And then they colored, well, for the average gene copies and whatever, and they colored here, selecting the genes that are you can look at the whole diversity of genes, or you can only select key genes that you know are representative of a specific metabolism, okay? Because to perform a complete metabolism, a complete path, you need a lot of genes. But there are some ones that are essential to perform it. So if you find that one, you can for sure say that that metabolism can happen, okay? So you don't need to look at the 20, 30 genes that are involved with that process. You can look at the one that is the key one. So you just select those, and you here they classified based on the metabolism they're involved to. So, and they cluster them based on their metabolism. So here, looking at the colors, which tells you the abundance of those genes, for example, you can see there are for example, here you have photosystem 1 and 2, so phototrophy, which is much more abundant. For example, that what is it? Nitrogen cycle. For example, you see that stripe, which is very dark. Those are all the genes involved in phototrophy, for example, which are very abundant compared to other one. Or the top one, which is aerobic respiration, very abundant. And then this is like very general information about metabolic like processes happening, not happening, what they could happen, because this is still DNA. Well, it could be just because sometimes, this is the, hmm? Yeah. Otherwise, the scale of colors could be like sometimes with heat maps, doing a log allows us to have a different scale of colors, which shows better the differences. Okay, I think we are done. Thank you.
Outside. There are gases that are completely inert, microbiologically speaking. The problem usually is that these gases are also pretty rare and are very expensive. For example, a great alternative would be to use argon. But who wants to pump an entire stuff of argon, right? Same goes for noble gases. We have plenty of noble gases that are non-reactive. But helium is rare and expensive, right? And the other one are only available in trace amounts. A good solution, generally speaking, is nitrogen, N2. Our atmosphere is 78, 77 point something % nitrogen. Microbes can use nitrogen, but only a small number of them does, and requires them a lot of energy to use it, to make ammonia and then use it as a nitrogen source. So they don't get energy from N2, but they need to invest a lot of energy to use a very small amount. So instead of using CO2, N2 would be a great alternative. Now, it's also not very reactive in terms of minerals and so on. It's not very reactive in terms of geochemistry. It's abundant in the atmosphere. I don't know how feasible it is to use it, mechanically speaking. But for sure, you know, if you pump CO2 with hydrogen down there, you're making a lot of people happy. Well, methane could be a good alternative because both methane and hydrogen are electron donor, microbially speaking. And so your community might be limited then by good electron acceptor to close your redox chemistry. So if you're providing two donors together, they don't cross-react, they don't have possible redox chemistry going on. It's potentially a good one. But you need to be aware of what microbes could be doing with methane down there, considering your mineralogy, considering other trace things that are present, and so on. Overall, I would say that we think subsurface environment are generally limited by oxidants, by electron acceptor, which makes sense. And so a lot of the things we worry about in microbiology, they're actually not happening because we are limited by electron acceptors. Other questions in general? David, where should we play with mud this afternoon? Probably outside, right?
How much is,
if it's not raining.
how much is-
No, we're not getting anybody dirty, if that's the question. Mixing mud with stuff and filling a bottle.
There are other rooms inside this building to explore.
Yeah, but we need some table space if we want to explore. The one downstairs could be good, the long table downstairs.
For the people which are doing the-
Or if it's not raining, we can do outside, which is probably,
Well,
it's raining now.
it's not raining now, but I know later, but in case you can use the room.
Yes, yes. Well, people can actually use this small desk they have in front of them. We just need to do something here and then they take it there. We'll see. So this afternoon, we start, instead of 3, we start a little bit earlier. We'll give you like a 10-15 minute presentation about the work we will be doing. And then we're actually doing a practical. And the practical is about you building one of these microbial ecosystem that you can take back home then. And these microbial ecosystems, they highlight very well concept of redox chemistry, cycling within the community, gradients that are created by microbes within an homogeneous structure, and things like that. And you know, within 2 or 3 months, you'll see how they develop. Yes, for your office.
It's a fantastic object.
Then after that, once we're done with that, we'll have 2 more talks from Martina and Gabriella on what we've been doing with natural hydrogen springs and wells, and what we've been doing in the lab as far as incubation goes. Lei adesso ne sta facendo in tubi di vetro sottili verticali sono delle bomboniere davvero.
Then just have to keep it there in my office with the light, which is with the rest.
Sì, si possono fare una bomboniera per quelli del corso di laurea.
Can you bring it on a flight? I think. Yeah.
Oh yeah, absolutely. Just put it in. You put it in your check-in luggage, and you're all done. Yeah, in a few months we'll get updates on the Shine. Any other question or yes? A what? Okay.
Can we edit the genes of the bacteria so that they can consume CO2 or produce hydrogen?
So theoretically speaking, yes. People, we can edit very precisely some genes. We can add or remove genes from organisms. We can even add entire pathways, which is many, many genes that work together to make something happen. This in theory. In practice, things get a little bit more complicated because there's a lot of interdependence between the different systems within one organism. So people have been trying to get E. coli, which is the most common microbes we use in the lab, which is an heterotroph. It uses organic carbon as carbon source and make it into an autotroph, somebody that can use instead CO2 as a carbon source. Things are not easy. People have been trying to get E. coli that can respire either oxygen or nitrate as terminal electron acceptor to make it into a sulfate reducer, make it use sulfate. There's a lot of genes that need to go inside and work together, and there are interdependencies that not always work. People also tried a different approach. They designed from scratch what they call a minimum genome of an organism, and then they synthesize it and they put it inside a cell that was empty without genes. And this work done by the Craig Venter Institute, and it worked up to an extent. So you are using a machinery that is already there, you're just removing information, you're putting there new information and let it run for a while. So theoretically speaking, it's possible to engineer things, to do things. Practically, there are constraints, there are technical difficulties. You need to well select what genes you're providing where, and that's for giving entire genes. Now, manipulating a single gene to alter activity, we are less good at it. It's more complicated because not always the exact biophysical mechanism of action is very clear. So there's a lot of trial and error. I know people that tried to engineer single genes that do something to make it at a higher temperature, for example, increase thermal stability of the gene. It's 3 to 5 years of work just to increase the thermal stability of one gene. So we are not there yet at doing it at the scale we'd like. But you know, in the biotechnological field, there are people that are playing with gene content to optimize production of biodiesel, or lipids to do esters to do diesel, or optimize bioplastic production, polyhydroxyalkanoate, polyhydroxybutyrate that are used to do bioplastic from bacteria. So people are playing with all this. I would say we're starting to tinker with these things. We're getting better, but we are not there yet to be able to engineer a full scale, something we want that does what we want. And then there are additional considerations to do. So many people are afraid of Frankenstein microbes, as they call them, or microbes that have been genetically modified, because once in the environment, they might, take over and be widespread. So you modify an organism. This has been discussed a lot in the context of petroleum degradation in the ocean. Oh, if we can engineer an organism that is very, very good at degrading hydrocarbons, whenever we have an oil leak or an oil spill, we just dump it from the top and it's going to do great. And many people were against this because they said we are spreading a microbes that is not part of the natural community. What if takes over? They're right, theoretically, but practical experiments show that whenever you engineer an organism, you dump it in the environment, it dies and people take over. Because people in the environment are better adapted to those conditions than whatever you can engineer. And so right now we went from releasing in the environment organisms that are being bioengineered to work better, to go to the system where the oil spill happened, isolate microbes that are already there doing the job we want, figuring out how we can help them to do it better, and then dumping them back in the ecosystem again. So it's called bioaugmentation, autochthonous. Take a microbe, understand how you can make it work better, and then go back to the environment and do it. Because we realize that many times when we drop microbes in the environment, the fear is that they're going to take over. The reality is that they get destroyed pretty quickly. No, microbes to do the job need to be happy and multiply. They need to become extremely abundant. Otherwise, whatever million microbes you dump in the ocean, that's a drop in the ocean compared to billions that are present in every kilogram of seawater, in every liter of seawater. So unless they're happy multiplying, they're not really doing the job you intended to. They're actually...
The experiment, what's happening is when they stop multiplying and they die.
Yeah, they never took over the community.
What is not triggering?
We don't know what it is. We know it's competition. So they're competing for resources and they're worse than the microbes that are naturally present there. It's normal,
So it's like when you got the lion which is growing on the zoo and then you bring it back on the wild system.
And it dies. Because it's not ready for the wild system. It's adapted to the zoo, right? We have opposites with macro organisms, big stuff, where you bring them there to do something and then they just take over and destroy the ecosystem. Cane toad in Australia. We have example of species that were imported somewhere, then they made an ecological mess. But we know much better the ecosystem of vertebrates. We don't really understand the ecosystem in which microbes interact. So we are still, I would say, blind to what their real needs are. That's the reason why we cannot grow them in the lab. And that's the reason we cannot predict how well they will work in natural ecosystem.
Yeah, they are simpler as structure than any other vertebrate.
As structure, but not as chemistry and biochemistry. Strictly metabolically speaking, every single animal on this planet does the same metabolism. Sugar plus oxygen equal water plus CO2. And every single plant and algae on this planet does the exact same metabolism. CO2 plus water and light gives you sugar and oxygen, period. You can write the entire metabolic diversity of plants and animals in 2 reactions, and you don't need anything else. Microbes, 1,500 reaction and counting, and we don't know what they're doing. So while structurally speaking, in structure and interaction, social interaction, direct organism interaction, competition, predation, we are very complex on the surface, shape, color, and size. Biochemically, the reason we can use a fruit fly as a model for humans is that, we eukaryotes, we share a lot of the same DNA and mechanism. The same genes that tell you how a fruit fly makes the head one way and the tail the other way, the OX genes, are the same gene that structure embryo in humans. We're not that far away. Microbes are on the other. All animal diversity fits in one line of the tree of life. There are hundreds. So yes, people tend to think that microbes, because they're unicellular, is more, they're simpler. But we need to define what you mean with simpler. Genetically and metabolically speaking, no, it's the exact opposite.
So first, do you have a same type figure for the population of the microorganisms to hydrogen concentration? And because it was just consumption, not the total population.
Which one? No, it's after. This one.
It says that the hydrogen concentration, it gives some places that the microbes doesn't care about hydrogen anymore. It's not a limit. But first, if we train this for population of microorganisms by hydrogen concentration, is it the same curve or?
Population, what you mean population? Like this graph. So for us, usually we measure substrate disappearing over time, right? You give hydrogen and it's going away. So you give hydrogen and hydrogen gets consumed in X amount of time, okay? So you have a graph that is different where things, hydrogen concentration goes down over time, right? You start with a lot of hydrogen and it goes down over time. Now, if you do this with different concentration of hydrogen, you give it them 1, then you give them 10, then you give them 100, you get different speed depending on how much hydrogen you give. Why? Because let's hypothesize that in this room, there are 10 people that can break down hydrogen, okay? And hydrogen is coming in 1 molecule at a time. As soon as it comes in, somebody's gonna break it. Now you can double the concentration of hydrogen and you still do it. You can reach up to 10, and we are all doing it at the same speed. If you then go to 15, the time that we can break hydrogen is not limited by the arrival of hydrogen, but how fast we are. Now, this is a situation in which all 10 of us have the same speed, right? Imagine that within the population, we have different speeds. Some people are faster at breaking the hydrogen than others. Usually, the people in biology, there's this trade-off I mentioned between affinity for the substrate and speed at which you use the substrate. So the people that get the hydrogen as soon as it gets into the door, they're very slow at breaking it. And the people that takes a little longer to get it, they're very fast and then pulling it apart. This is because hydrogen needs to enter a place where it gets stuck, right? If you are very sticky, you break it, it's going to take a while to release the problem. If you are not very sticky, you're less precise in your work, but you break everything you get. So your speed at getting hydrogen is lower, less sticky, but you break very fast. Now we do this experiment of hydrogen going down over time against time, concentration against time, for multiple concentrations usually. We do it at multiple concentration. And then we plot a plot like this, where we have the speed at which the hydrogen is used against the concentrations. Now, different population of microbes will have different microbes because they're different, and different population of microbes and different enzymes will have a different slope of this curve, right? Some of them will saturate very, very early. Some of them will saturate very, very late. Some of them will have different shape of the curve. Now, this curve is telling something, is telling us something about how fast and how efficient and how the population is and how much inhibition starts to be above a certain concentration. You can increase the amount of hydrogen, you never get faster than that in rates, okay? You reach a plateau. The two real parameters that we look for are something that is called the concentration at which you reach 50% of your speed. It's called alpha V max. In biology, that's a very important modeling parameters because it tells you that parameter you can use for kinetic modeling, concentration of the speed, and gives you also what we call the affinity of the enzyme. How good the enzyme is at getting the substrate. The higher the affinity, usually, the higher the affinity, the lower the speed. And so we do this type of experiment, we get this type of curve to get two numerical parameters that we use them for modeling of populations. Now, the problem is that this work is usually done in a system where you get a microbe, you grow it in the lab, you kill it, you extract, you purify the single protein that is doing this work in milligrams amount, and then you can run these experiments. Running this experiment in one microbial culture, they might have different enzymes inside that are competing with each other, gives you a bulk microbial. I don't think most people do this experiment with full populations. Why? Because during growth at different concentration, population will change. So if we are 10 people here and we don't change in ratio, we could theoretically get parameters for the population. But in microbiology, if you give little hydrogen or more hydrogen, people that are good with little or good with lot will grow. And so you're getting a changes in population that change the parameter over time. So you're getting a very distorted view. Now, I'm not aware of many work that has done this on complex population. But that's definitely one of the direction we should be going to get two parameters, which is maximum theoretical rates at which the microbe population can use the hydrogen, and modeling parameter for the speed, for the integrated rates you can use in thinking about time, for example. And from a graph like this, you can also get the inhibition concentration that you start with. I don't know if that answers your question.
When we increase the concentration of hydrogen, it also means that we lower the concentration of other stuff.
Yes, in the gas phase, not necessarily minerals and other things.
Okay, but so can we say that, like in underground hydrogen storage, which is mostly the gas phase and solid, like in the places that hydrogen is the maximum concentration, the microbial activities are in their minimum?
Well, so if you imagine a salt cavern where your volume to surface ratio is dominated by a very large volume and not a long surface, okay? The only place where microbes will be able to find water, even if it's a saturation of NaCl, an environment where to attach it will be essentially on the walls of the cavern, like on the surface of the cavern. Now, in that place, you will get a gradient of hydrogen to brines that are on the floor, or the liquescence of water, humidity that is attached to the cavern, or a small fracture where you may have things in. In that place, I think there is enough ratio between the different things for microbe to live. If you're moving inside the volume of the cavern, where you have only your, let's say, hydrogen and methane, no, you're not. Okay, there's no space there. The biggest problem we think with microbes is aquifer and porous reservoir, right? Where pretty much everywhere you have space for microbes to attach and create a small microenvironment. One thing we did not discuss is the ability of microbes to make what we call biofilms. Like plaque on your teeth, wound infection, the green if you don't wash very, very well your sink or your outdoor stuff. All the slimy little layers there are what we call microbial biofilm. Microbial biofilm often are made by multiple population of microbes on top of each other, where they exchange nutrients and there are huge geochemical gradients inside there. So people at the surface use the hydrogen, and then everybody down there uses byproduct of the metabolism. These things are very, very known attached to rock and sand grains and all these kind of things. It's actually one of the main ways microbes live in the environment. So in porous media, I will not be surprised if microbes grow to make biofilm and actually clog the pore space with their own body, just building bridges. And I'll show you this afternoon how big and how thick a biofilm can become, just before we do our little practicals. But we're talking about cells that are 1 micrometer or less in dimension can build biofilm that are 20 to 30 centimeters thickness. So you walk on it, literally. You don't realize it. Yes. Yeah, that's the absolute upper bound, considering what? A monolayer attached to the cell, but that's not the upper bound, because when they organize in biofilm, they're not monolayer, they're multi-layer. So what should we do? 100 micrometer thickness biofilm? And depending on the porosity, that means full clogging, depending on the porosity of your, if you are in a sandstone, no. If you are in a mudstone, probably 100 to 150 micrometer, you are completely clogging the pore space. So it's theoretically possible, we can try, but again, what's the worst case scenario? Centimeter thick biofilm? You can make it as bad as nothing moves. The other thing is that we have no idea how the biofilms are structured in the subsurface. We have very little ideas about this. We have a few reports of biofilm from fractures that have been drilled into in some location globally. So you drill, you find a fracture within the core, you go there, you find biofilm attached on both sides of the fracture. But we have less ideas about this in porous media. There are some micro CT that have been done that they show there might be organic matter attached there. But from a micro CT demonstrating that that organic matter is actually a microbial active biofilm, there's a lot of work that need to be done. But theoretically, yes, you can calculate the available space for life in the porous media, which is the same available space that you have for hydrogen storage. They match 1-to-1. And then you can try to subtract a fraction of that for biology to live in, 1%, 3%, 5% of your porous space. And then you can, theorize from one of these kinetics. If everybody there is using hydrogen, worst case scenario, how much hydrogen we're talking about. But worst case scenario are always very scary. And I'm not sure that they're representative or useful in this context.
Can you repeat what you said before? Biofilm tend to grow better in months than...
No. I didn't say better.
Just saturate faster.
Well, if you are in a place where you have a very, very thin pore space, biofilm might clog it faster than when you are in a coarse, very porous media, right? There's more space to be occupied.
You can use them to create impermeable. You impermeable.
Well, semi-permeable, because gases, gas, microbes don't make tight spaces unless they start to precipitate minerals in the biofilm. And in many biofilm, microbes like to precipitate calcite, aragonite, and things like that.
Which should impermeabilize.
Theoretically, yes. For example, we are discussing with a colleague the contribution that microbes might provide in creating hydrothermal seals in volcanic system. There are good chances that some of the sulfate alteration clay minerals they create hydrothermal seals in volcanic system might be facilitated by microbial activity in oxidizing reduced sulfur species, and therefore providing sulfate and sulfuric acid that reacts to create the sulfate clays. But this is, I would say, this is pretty frontiers. There are some colleagues that are very, very good in biomineralization that could be interesting to involve in this. And we did study it very, very well in context of stromatolites, for example, where that's biomineralization. Biotic mineralization in stromatolites is a big portion. Well, some stromatolites, but the living one we have on Shark Bay in Australia, for example, or the Bahamas, they're all living stromatolite where microbes play a huge role in calcifying. Yeah, that's... And the layers there are just different. So this is a microbial mat in the flow of a hot spring. So hot water was flowing on top. The top is green, which means you were under light, and there are likely phototrophs there doing oxygenic photosynthesis. CO2 plus water equals sugar plus oxygen. And people underneath, the darker color, they're probably using some of the compound that are passed around. And the red one might be either pigments, carotenoid to protect from the heavy UV, or maybe iron oxidation, because they're using iron too from the spring water as an electron acceptor. And if you look at the finger, that's probably a 1.5 centimeter thick biofilm, and it grows in weeks. It doesn't take years to grow.
Have you encountered any biofilms that are from the core, that are taken from the oil and gas fields?
I never encountered them. I did experiment of biofilm formation in wells that are 100 meters deep, 100 meters deep, and things are pretty slow down there. Exactly because you are limited by electron acceptor, electron donor, or organic carbon, and so on. So unless you're stimulating things, things are slow. I can say, however, that there's another type of biofilm we have not discussed that you don't see here. So this is a thick mucilaginous biofilm, grows layer on top of layer, like they grow on top of each other. There's another type of biofilm which we call endolytic biofilm. These are microbes that grow within rocks, usually carbonate rock or sinter, silicate rock, and they grow just under the surface. So the outside of your carbonate is completely white. If you break it below 1 millimeter, you start to see layers of colors. And they dissolve partially the carbonate there, and they grow there because they have a shield against strong UV light, but light is still penetrating under. And they make layers up to 3, 4, 5, 1 centimeter thick inside the rock layer. And pretty impressive, if you ask me, because in that case, they're altering the rock layer structure. Yeah, but we did experiment where we broke a carbonate layer in a calcite precipitating hot spring, where you are pH 8 and saturated in CO2, and there's a lot of calcium, so calcium carbonate naturally precipitate. You break the place, you come back a month later, the paste is reconstructed, the biofilm is there, is very happy. So we're not talking years, we're talking weeks, days, months, which is within the time frame of storage activities. The problem is always when we are thinking about these things in hot springs and surface spring and near surface environment, these are high energy environment, right? You have plenty of organic sources and light, energy from light and oxidants and everything. In hot spring is even better. You have reduced compound coming up and oxidants at the top, is just smashing them in the highest possible energy place of this planet. When you are stuck down there, things are different. You get less supply of things. In a porous reservoir media, unless you have aquifers moving through, you are near fracture, things are limited by diffusion, unless you are actively injecting and pumping out. And diffusion through a porous system, it's not that fast. So things are much slower than what we can think on the surface. Other questions? Right now, the fastest microbe we can grow in the lab is potential skin pathogens that can attack also your muscles and things like that. And is one of the microbes that is responsible for what we call septicemia, which is a general body tissue infection that requires you to cut one of your arms or leg, or you die within hours. Now, there are several microbes that can do this, but one of these microbes can double in population size every 14 minutes. So once the population is off an initial step, just explodes. Within 3 or 4 hours, you've done 8 to 10 generation times, and it's just super fast. Now, these microbes is generally associated with skin, member of this Corynebacterium, generally associated with skin. It's an opportunistic pathogen. So you have it on your skin everywhere. In hospital, you find the version that is resistant to antibiotics, which people commonly call the lawnmower, for how fast it can chop people down and kill people that are immunocompromised in hospital.
So it's true that the hospital is a very good place where to get.
Yes, absolutely. But this microbe is an heterotroph. So it doesn't build many things. It uses your protein, your lipids, your sugars, everything on you. On the other end of the spectrum, we have microbes that get only inorganics from outside, right? And they're autotrophs. They build everything. There are some microbes that from the outside, we only get sulfate, ammonia, CO2, hydrogen, and trace elements. They don't like, if you give them sugar, they don't take it. If you give them amino acid, they don't, any organic carbon, they will not touch it. And these microbes, they build everything from scratch, right? They don't repurpose lipids and sugars, and they can still double in size in 30 minutes. So they're a completely autochthonous industry where everything is built from the screwdriver to the nut, everything is made inside, and they only take double the time of an industry that imports everything from outside super fast. That's very impressive. So you need to think that some of the things we're discussing theoretically might happen very, very fast. Now, we don't know how fast in the subsurface, but lately we were in the field with Flavia. We collected sediments and waters from a hot spring. Sediments were black, water were transparent. We collect these to try to do microbial isolation and cultures in the lab. We drop it in a...
How much deep was the sampling on the hospital?
So we sampled the spring at the surface while they're coming out. We don't know the depth. It's theoretical depth with the gradient depth. It's transported up.
So you don't know it's collecting stuff, why it's not.
No, we don't know. But we know there was not too little mixing with the surface stuff because we track things like helium-4 and neon-20 and carbon-14. So we have proxy to know how much surface mixing or near surface mixing. So we know they're deep. We can calculate because of aquifer equilibration, geothermometry, geobarometry, and heat flow. What's the depth? Hundreds of meters to a kilometer in the area where we were, pretty much. So where things get back up, we close them in these vials, gas tight, anaerobic. We put it in the dark in a bag where there were ice to keep them cold. Within 2 to 3 days, water was clear, sediments were black, you pull it out, everything is purple. Seriously purple. And that's a microbes that we call an oxygenic phototrophs. They use hydrogen and H2S. They found the perfect condition to bloom completely. And everything became intense purple. I can actually show it to you. I have it here. When I say purple,
In the hospital with 150 degrees. Fluid, not to go to the hospital.
purple. Like purple, in 2 days. We were not giving the right condition. This was not us trying to isolate it. It was, somebody that was there in very low amount found whatever condition we were providing him to be perfect, to just bloom and occupy the entire space.
But when they blew, then they killed the other one.
Well, they compete for,
they
not
compete-
necessarily kill, but they
just
compete for resources.
Get out of the food.
Somebody becomes better at doing whatever job is available right there. Until they die, they run out of resources or virus kill them.